Okay. So, hello everyone. Good morning, good evening. And today we have a treat. We have now here Dr. Burkhard Becher from University of Switzerland, and give us a talk about the the immune response in neural inflammation. Just to give a brief introduction of Burkhard, he is a immunologist, particularly interested in inflammation research, in nervous system, and then he is also the director of the Institution of Experimental Immunology and the University of Zurich in Switzerland. Burkhard got his undergraduate in University of Cologne in Germany, particularly specialized in molecular genetics and biochemistry. And then further, he pursued his graduate study in McGill University in Canada, and to train in neuroinflammation with Jack Antel. His work focused on the role of microglia cells as brain-resident myeloid cells capable of instructing self-react T cells in the context of autoimmune neuroinflammation. And then in 1999, he joined lab of Randy Anolio at Dartmouth Medical School to extend his work in vivo models of chronic inflammation and in transplantation immunology using innovative transgenic model tools. So, his lab actually focused in the cytokine and how these molecules permit cell-cell communication between immune cells. Particular in 2003, he was recruited as assistant professor to University of Hospital of Zurich, where he continued to define the cytokine network in inflammation. In 2008, he became full professor and chair at the Institution of Experimental Immunology. And then he was awarded in different awards. For example, 2010, he got awards as Professor Max Kotana Award. 2019, he got Solberg Award for MS study. In 2020, he got ERC Advanced Grant. So I think Burkhard work mainly focused in neuroinflammation. In particular, he actually has extensive study in multiple sclerosis study. And then from my personal point of view, he actually, the work he published actually hugely influenced my early stage of career, particularly my PhD study. A lot of work he published, particularly of the dendritic cell function in multiple sclerosis, is really an inspirational work for my study. I would suggest to you all guys check out his work early, like 2010, 2005. So I think the other thing I want to particularly emphasize is he's really a very nutritious scientist. He has 19 postdoc and 40 PhD students who all actually move on to become very successful in academic career or leadership in the field. So without further ado, we are very much looking forward to talk. Please carry away.
Thank you so much, Juan. Thanks for having me. And when Juan asked me, he sent me an email saying, "Hey, can you give a talk at our online meeting?" And first he asked, "Do you remember me?" And of course I do remember. I do remember him very well. I did follow your career, and I'm quite impressed, of course. You've done marvelous, and it's a pleasure to reconnect, and it's a pleasure to be here today. And if you don't mind, I will zoom you all away. Otherwise, you're in the middle of my video where I don't of my image here, and I always get confused with the Zoom talks when I suddenly can't read a title or something like this. And as Juan said, I would like to talk to you today about a subject that I find particularly interesting, which is how monocytic cells or monocytes, which live in the systemic immune compartment, move into tissues and then become what I would call a phagocyte, and how they contribute to tissue inflammation. And so let me start with basically with this image here that has basically, it's an analogy, obviously, because I'm not an astrophysicist. I don't know much about astrophysics, except for the fact that we are all aware of the fact that somehow this is the moment the universe was created, the Big Bang. And we know that the universe has expanded, has expanded, has expanded. We're still expanding our universe. There's a way to see this has to do with the redshift of our of distant stars. And again, this is not about about this. But the question is then: is there going to be a maximum expansion, or will the universe continue to grow, or will the universe start to close again, and we have another big moment called the big crunch, and the end of all time and everything in the universe? So that's the question astrophysicists have, and they don't have an answer to this, but I have a good immunology analogy to this, which is basically one of those things that have changed the course of nature and immunology. And that happened in 1989 in a publication in the Journal of Immunology, which when I was a kid was basically the big stuff. That is where you get published when you do something really outstanding. And this is like the guild journal for immunologists. Things have shifted a little bit for this journal and for many others as well. But nevertheless, this paper by Tim Mossman and Bob Kaufman basically describes that helper T cells come in different flavors. You have Th1 cells that make interferon gamma, and you have Th2 cells that make interleukin-4. And this has changed the language of immunologists, because now we talk about things like type 1 immune responses, which are specifically geared towards intracellular pathogens and cellular immunity, and type 2 immune responses against extracellular pathogens. So this has basically initiated a whole new set of research into, well, when is something a type 1, when is something a type 2 immune response, how are they being described? And then also this crept into the subject of tissue inflammation. And along the way, the reason why I bring this analogy, the universe has expanded. I pulled this off Wikipedia, or just Googled it somewhere. I saw this image and I see new entities. Obviously we heard about regulatory T cells, which are pretty much hardwired to the expression of FOXP3 to be regulatory T cells. But then other cells like Th22, Th17, Th9, T follicular helper cells. And then recently I saw this article, which I actually thought is a really, really good review paper on the multiplication within only one subset alone. These so-called Th17 cells will make now there exists as Th917, regulatory Th17, Th217, Th117, and of course XTH17, etc., etc. And then there's of course a connection to this Th22, and it became super, super complex. Again, the article is brilliant. It's really well written and well researched, but I have a problem personally with the nomenclature and the further expansion of the T cell universe, because it doesn't serve any purpose anymore. We don't learn anything from this, in my opinion. And again, it's an opinion piece, and a couple of my friends share this opinion, and so we put out this review article, which I urge you to have a look at if you like, that basically says, look, let's go and categorize T cells based not on their phenotype, but on the function. Function meaning, when helper T cells, what is the primary function of a helper T cell? I just pronounce it this way because obviously it's help. And we think that type 1 helper T cells are primarily well equipped to help mononuclear phagocytes, such as monocytes and macrophages. And the cytokines dominated, and we'll talk about it some more, interferon gamma and GM-CSF. And then you have type 2 responses that include, for instance, what happens in follicles, so the emergence of B cells, B cell maturation, but also an allergic response, maybe eosinophils and mast cells. And these are what we call type 2 immune responses, mediated primarily through a set of cytokines here that is often produced together. And then you have these type 3 immune responses, and they are geared to interact with non-immune cells. So type 3 T cells help non-immune cells. And for instance, these cytokines IL-22 and IL-17 are particularly good in interacting with tissues of the body lining, such as the lung and the gut. And this is where this is very prominent. And these are epithelial cells, for instance. And we think that this categorization is a little bit less rigid and a little bit more interesting, and allows us to actually look at function rather than phenotype. Because if you find a T cell, because I'm always struggling, what is a T cell that makes, IL something, TNF, IL-10, interferon gamma, and what have you all at the same time? What do you want to call that now? You know, in the end, they're all Th beta actin cells, if you wish, and that may be too minimalistic. So we think that this is a decent compromise. So check out this little review. It was fun to write. I've been thinking about this for a long time. I think it's just something to ponder and discuss, and maybe discard if you don't like it. Either way is fine. Having said that, so one of the questions then were asked by these two gentlemen here, this is Florian Ingelfinger and Eduardo Galli, was what, if there is any, is the immune signature in multiple sclerosis? Can we identify that? And what they did is a relatively simple experiment. They took samples of, and these are peripheral blood samples of patients with multiple sclerosis, of patients with other, with non-inflammatory neurological diseases. That would be headache, for instance, chronic headaches, and patients with other inflammatory diseases such as rheumatoid arthritis, lupus, etc. And so these samples, a lot of samples were taken, and they did a relatively simple study based on the fact that we purchased the Cytokine a while back, and then they were able to look at 84 different parameters, including 14 different cytokines, chemokine receptors, etc. And the samples, we developed a method to barcode those samples so we can put 120 samples together and then stain them with the different antibodies for cell surface markers and intracellular factors, and then run the Cytokine analysis followed by algorithm-guided analysis. Now, the algorithm-guided analysis then was pretty hot and novel and something that wasn't done extensively for. We used to use we used to gate and follow our hypothesis. This here is not hypothesis driven at all. This is a blind experiment. You call it a fishing expedition if you wish. But the use of computers has tremendously helped in allowing us to have fruitful fishing expeditions. Anyhow, so what came out is that in patients with multiple sclerosis, there's a small population of cells that sticks out. Sorry, I have to wait 1 second. There's a spider on this wall and it's driving me mad. The spider is no longer on that wall and I'm feeling better. So there's these cells here, this population that sticks out as being elevated in patients with multiple sclerosis happen to be CD4 positive. Here's basically the numbers. This is the analysis was run. It's called CELL-CNN. It's a very interesting algorithm that even looks for rare populations which may be dysregulated. And what you'll see next is that it's elevated in patients with multiple sclerosis and clinically isolated syndrome. I skipped this here. The signature of these cells, a little bit more detailed, is they make interferon gamma. They also make GM-CSF, so they are type 1-ish cells, and we'll come to this later on. They also make TNF and IL-2, and they are expanded in the cerebral spinal fluid of patients. So in other words, we were asked, is this a pathogenically relevant population? And the answer is, I don't know. I still don't know. But I can tell you at least that patients who have multiple sclerosis will have many, many of these cells, of this population of cells invading the central nervous system, suggesting that this is a shadow, what we see in the periphery is a shadow of the inflammatory response in the CNS. And further supporting this is the fact that, as you can see here, patients that are treated with an oral immunosuppressant, in this it's called dimethyl fumarate. It's a relatively effective drug to treat multiple sclerosis without having to be injected every day or every week. You see a decline of this population in the treated group. And we know now actually in the treated group of patients that respond well to the drug. The non-responders, and it's not part of this study here, the non-responders do not show this decline in this, let's say, MS signature. Having said that, that's just basically the introduction to something that I find rather fascinating, which is the presence of these type 1 cytokines. And the one type 1 cytokine I personally love the most right now is called GM-CSF. And I'm asking the question, is this pathogenically relevant? Now, first, something about GM-CSF, it stands for the granulocyte macrophage colony stimulating factor. And you should know that GM-CSF is functionally not a granulocyte macrophage colony stimulating factor at all. In the steady state, it's produced at very, very low levels, and it's almost exclusively expressed or maintained in the lungs, because you need GM-CSF for the development and the maintenance of lung macrophages, of the alveolar macrophages. You have also a minor effect on the CD103 positive CDC1s across epithelial tissues. But in the absence of GM-CSF, you still form perfectly fine and normal conventional DCs, macrophages, and granulocytes. So it is rather redundant as a colony stimulating factor. It's not that in the steady state, mind you. In inflammation, things change dramatically. It's obviously required for neuroinflammation, and I think Joan was referring to some work a while back where we basically found that GM-CSF is needed by helper T cells, and it's required to cause neuroinflammation. We found that the primary source in EAE are indeed encephalotogenic T cells. They make a lot of GM-CSF, and that dysregulation of GM-CSF can trigger brain inflammation alone. And this is in an autoantigen independent fashion. So we don't induce neuroinflammation. We just dysregulate GM-CSF and we see brain inflammation. And that this molecule can activate the transition of monocytes into what we call inflammatory tissue macrophages. So as such, GM-CSF is really, really the primary communication conduit for pathogenic T cells to talk to mononuclear phagocytes. And oh yeah, no, here's the data. I was wondering why I'm citing this paper here, which I'm going to show you quickly. So as you remember, in multiple sclerosis, one of the signatures is elevated levels of GM-CSF. And together with a PhD student, we were wondering what happens if you dysregulate GM-CSF in mice. And so Sabine Spath made this knock-in into the ROSA26 locus using zinc finger nucleases. Actually, mind you, this is before CRISPR-Cas, obviously, where basically a strong promoter drives the expression of GM-CSF and GFP, and it's driven by Cre recombination on the CD4 locus, but with tamoxifen, so it's inducible. So what happens is when you give this crossing, when you give the mice after they've been crossed, when you give them tamoxifen, CD4 positive peripheral helper T cells will start making GM-CSF, and you can dose it. You can give it very little, you can give it a bit more tamoxifen, you give it for longer time, and you can therefore change the number of GM-CSF dysregulated T cells. We think this is a we thought it was a relatively clever way to model GM-CSF dysregulation that we see in multiple sclerosis. And what happened is, and I have to just do something now because yeah, okay, so here I have to go here, automatic. Yeah, now this should work. What was happening is actually nothing for the longest time. These mice were perfectly fine and showed no phenotype. So Sabi was giving them and feeding them through oral gavage as well, in all kinds of different ways. We gave them tamoxifen, and nothing much happened. The mice were fine, and I asked her to get rid of the strain because it just didn't work. It was a silly experiment that failed. But then, like everyone else in my lab, Sabi did not listen to me and decided to keep those mice and continue me paying for those mice. But what happened after about 10 weeks, I was just too impatient. She was right, I was wrong. After about 10 weeks, you can see this phenotype here. The mice actually developed this very strong disease phenotype. Obviously, this we're talking about neuroinflammation. The mice got horribly sick and we had to sacrifice them. And the penetrance of this disease is incredibly high. It's virtually 100%. So you give tamoxifen 0, and about 10 weeks later or so, the mice start to get sick, and they all get sick. As you can see, there's probably one outlier, and that's it. Everybody else got sick. And without, yeah, I've showed you the wrong slide first. I apologize. Let me show you this first. What we saw is inflammation of the brain. Essentially, what happens is into the brainstem, the cerebellum, you saw this rotatory phenotype that's indicative of a stem and cerebellar invasion or inflammation or dysfunction. We also see changes in the meninges and the spinal cord. We see this inflammation and a massive invasion of these cells here. This is the most prominent population moving in, and here we call them MODCs. They're basically monocyte-derived cells. I'm very agnostic about the nomenclature, and this is the last part of what I'm going to talk about to you today, whether we call them MODCs or MOMACs or monocyte-derived, what have you. We call them monocyte-derived cells at this point because we didn't want to commit to one or the other. But you can see that these cells, when they invade the brain, start eating away the myelin and destroy actually quite a bit of myelin, and that's the reason why these mice are so sick. Mind you, this also works in when all the T cells recognize ovalbumin and nothing else. So in an OT2 background, this also happens. This is not autoimmunity driven by an autoantigen. This is preferential invasion into the CNS. Here's the summary of this in the graphical abstract, if you wish. We induce the secretion of GM-CSF. We see an expansion of inflammatory monocytes, and they do not invade much the joints, the skin, the colon, the pancreas, but we see some of these cells moving into lung, liver, and kidney, and the CNS. So the difference is their profile. In lung, liver, and kidney, the cells look like activated monocytes, if you wish, but in the brain, we see a massive increase in the production of reactive oxygen species, production of interleukin-1, and the ability to phagocytose. So the gene expression profile of the cells in the CNS is fundamentally different from these other tissues. And then I think I'm going to quickly talk to you about the work of these two, which is this is Selma Tuslak and Juliana Komitsky. Selma is a postdoc, Juliana was a PhD student in the lab, and is now moved on like everyone else into industry, making lots more money than everyone else, probably also more than I. And they made a fate map and reporter of GM-CSF mouse by knocking into the GM-CSF locus this construct here. So this is now with CRISPR-Cas. So you have the enhanced recombinase, the iris, and GFP. So, and this was then into the locus of the GM-CSF, into the GM-CSF locus, meaning that the GM-CSF promoter is now driving this construct. And so the resulting line, we called it the fate map and reporter of GM-CSF. And because it was too long, we took the acronym FROG. So F-R-O-G, frog mouse line. And so I show you a lot of frogs now, not because we work with frogs, just because it's easier to explain what we're doing. So if you take frog mice, this again a mouse, and this is the construct, when GM-CSF is being produced, the cells automatically turn green, right? So because our construct forces the production of EGFP. Interestingly, however, when we cross this mice with a fate mapper, what happens is that every cell that makes GM-CSF will now turn red and green simultaneously. So GM-CSF producing cell will be green and red, and the cell that has made GM-CSF will be red. So the GM-CSF, or the red cells, have had made GM-CSF and or their progeny of a GM-CSF producing cell. And this worked actually surprisingly well. It's not that most of our constructs work, most of them don't, but this one worked really, really well. And then we looked into who's making GM-CSF, and you can see it here in the lymph node. When you look for red cells, and let's focus on the red more than the green, the signal is so much better, of course, is NK cells and CD8 T cells and gamma delta T cells, but also a few CD4 positive T cells. So I was hugely disappointed by this. I did not expect that to happen. Like NK cells, that weren't important, and CD8 T cells can't be important in the context of an EAE that is induced by an MHC class 2 peptide, right? So MOG is a 20-mer, and it stimulates CD4 T cells and not CD8 T cells. However, when we look in the brain, and this was hugely gratifying then in the central nervous system, we find indeed that there are CD4 and CD8 T cells invading now the brain, but the CD4 T cells are the ones that are still making GFP. And it's very simple explanation is because they see their antigen. The antigen is in the brain and they see it there, and so they're being reassured that this is the right spot, and antigen and TCR engagement drives the production of the cytokine in the brain. And then we could now, the construct, however, is a little bit more interesting than just a fate map and a reporter. It's more than, it's beyond the frog. We can do more. We could, for instance, we decided to cross the frog mice with ROSA26 DTA. And so what happens is every time GM-CSF is produced, the cells that produce GM-CSF will immediately commit suicide. So we call them the suicide frog mice. And all the GM-CSF expressing cells, once they start making GM-CSF, will die, will commit suicide. And you can see here, this has a major impact on brain inflammation, on neuroinflammation. It's basically dramatically reduced. The mice are barely getting sick anymore because you do need these cells. These are the most important cells. But what was interesting to us is that you didn't need any other cells. So the number of T cells invading the brain was unaltered. The same number of T cells going in, whether you have GM-CSF producing T cells or not. The difference is, in the absence of GM-CSF producing T cells, other T cells go in, but they do not lead to the invasion of these monocyte-derived macrophages, so monocyte-derived cells. Those are dramatically reduced in the absence of GM-CSF producing cells. And the other cells that are coming in are making cytokines such as interferon gamma and IL-17 a lot. There's lots of IL-17 producing cells now coming in, but that is not compensating for the loss of these GM-CSF producing cells. So let me just look at the time. We're looking good. So take-home message is tissue inflammation, no matter where you look, is always dominated by myeloid cells. It's almost never, or I don't know of a disease where you have more lymphoid cells invading the tissue. I hope I convinced you that GM-CSF is able to license monocytes to mediate immunopathology. And the loss of GM-CSF expressing cells, in spite of the presence of interferon gamma and IL-17 producing T cells which expand, still halts CNS inflammation. And the rest I didn't show you, so why should I talk about this? And I show some work that's we resubmitted yesterday, but it's accepted, and that has to do with this thing that happens to monocytes when they see GM-CSF. So I talked to you about this a lot and say you do need, and Andy Croxford a few years back showed that when you delete the GM-CSF receptor in monocytes, the whole system collapses and you can't get EAE anymore. So without monocytes sensing GM-CSF, EAE is gone. And yet, we still, until today, do not really know what GM-CSF really does to monocytes. And so let's look a little bit, if you don't mind, in the last few minutes at the story of how monocytes actually emerge. They emerge from the bone marrow, and where we do have a population that we like to call Ly6C high monocytes. In mice, in humans, they exist as well. I don't know the markers by heart right now. I think it's 14, 16 positive. And then you have Ly6C low monocytes. They exist in our peripheral blood, and these are blood patrolling monocytes. The function of them is still a little unclear, and these are not the cells we talk about right now. They're not really involved in tissue inflammation. These guys here are the ones we focus on right now. So through the chemokine receptor 2 and CCL2 and other molecules, you can get an engagement of these cells getting out of the bloodstream and moving into the tissue. And here I show you this is supposed to be a brain. And in the tissue now, they will encounter T cells which produce these type 1 cytokines, as I showed you in the very beginning, predominantly interferon gamma and GM-CSF. And now we have this complicated thing that there are monocytes invading the brain, but they change and do what we call, and I call it from now on MTPT, the monocyte to phagocyte transition. And so what was described in the past, and a lot of people are fighting about this. It's very interesting how the phagocyte folks can get angry about nomenclature. So we have these MHC class 2 expressing cells that often people call monocyte-derived dendritic cells, MODCs, because they have lots of features in common with dendritic cells. Or you can see these MERTK high positive cells, which a lot of people say are more macrophages. And there's a big discussion as to whether whether this cell is an end. So actually, here's the question. I have it here, I think on the right. Hold on. Yeah. So the question is: are monocytes the precursors to MODCs and MOMACs? That's one hypothesis. The other one is that monocytes become MODCs and then become MOMACs, that this is just a transient state. That's one hypothesis. And the third one is that maybe there are two kinds of monocytes. Monocyte precursor 1 gives rise to monocyte-derived dendritic cells, and the other precursor gives rise to monocyte-derived macrophages. And to test this, Donatella and Anna, who I showed you just in the beginning, we're taking these mouse strains, one that we made ourselves, which is the CCR2 CRE-R, treated with tamoxifen, where you get a very good labeling of monocytes, but also labeling of pre-disease. And then a mouse made by a buddy of mine, Florent Ginoux, he made this MS4A3 Cree mouse as a constitutive Cree line, where you basically label the GMP in the bone marrow and the CMOPs to almost 100%, leading to a very, very high level labeling across Ly6C high monocytes, but also across Ly6C low monocytes. But these are the ones, again, that we were interested in. And so these two lines were enable us to fate map the cells. And the idea was to use both lines simultaneously to induce experimental autoimmune encephalomyelitis. And at the onset, about a week later, sacrifice the mice, but also sacrifice mice at peak disease. And looking at, so here's basically the scheme, and follow the monocyte from the moment of immunization over disease onset into peak and see how they behave over time. That was basically the idea. And then using these two fate mapping systems to actually join the information together and to have a more definitive answer as to what is the feature or what is the fate of monocytes in an inflamed brain. But at the same time, we were also interested, while we're at it, and we have to sacrifice mice for this purpose anyway, not just to look at the brain, but also look at the draining lymph nodes. And so what happened is, at these time points, the brain, including the brain and spinal cord, were taken out, and the lymph nodes were taken out, which is where the priming happens. And then we made a single cell suspension, and on the one hand used high dimensional spectral flow cytometry, but also single cell RNA sequencing to tease out some more details that we didn't have with one or the other method. And so one of the things that we found very early on, very simple picture I'm showing here, is that we see this sort of I'm assuming here a trajectory, but these are basically monocytes, and then we see over time the expression of MHC class II molecules, and then later on we see more macrophage-specific markers such as 64 and MERTK. And looking at all these markers together and just running a very crude pseudotime alignment, we found that monocytes first, so in the non-immunized mice, there isn't much happening with monocytes. You do see these populations obviously in the central nervous system, but they are exceedingly rare. There are almost no monocytes in the brain, so it's really blown up. But in inflammation, we see monocytes slowly disappearing and moving into these, that's what we call monocyte-derived dendritic cells. And in peak disease, we see more of these monocyte-derived macrophages, suggesting, when I told you the different hypotheses that we had, that maybe that MODCs are a transitional state, suggesting the data is not very solid here. This is just a phenotypic look at what's happening here. I'll go into a bit more detail here. So basically, I told you one way of doing this in a bit more sophisticated way is to look at at RNA velocity, basically looking at splice variants, and to assume by that analysis what cell is a precursor to which cell to which cell. And when we do this from various tissues, in this case we include bone marrow, the blood, and the central nervous system, you can see that there is the precursor population, obviously in the bone marrow, that yields then back into these Ly6C high monocytes. In the blood, this is the most prominent population, Ly6C high and Ly6C low monocytes. But in the central nervous system, we see these populations of monocyte-derived dendritic cells and monocyte-derived macrophages. And so this basically is the movement of the cells that basically suggests that our initial assumption that this is a transient state may be correct. Go into a little bit more detail here, and actually not too much, because I don't think it's important, and the paper will come out soon together with all the additional details. We again could show by looking at velocity and also using PAGA here that we do see a movement of monocytes through a gradual education across MHC class II antigen presenting cell features of the MODCs into what we call the MOMACs. But the more interesting question for me, again, this is phenotype. This is all very nice in my opinion, and really telling me much about this. But I also wasn't really worried too much about this question, whether there is one or two precursors and so on. I find it academically very, very interesting. But what as my passion is cytokines and how cytokines regulate this behavior in the central nervous system, in the inflamed brain. And so again, I told you T cells that invade the brain will make these two cytokines, GM-CSF and interferon gamma, predominantly. And monocytes will sense both GM-CSF, interferon gamma, both at the same time. And what is the effect of that? And can we tease this apart? And what matters and what doesn't matter? And so there is a couple of experiments we felt were interesting to do. And then we realized many of the things we want to do can't be done. In particular, because a mouse, a monocyte that does not sense GM-CSF, so this is a GM-CSF receptor deficient mouse, will be EAE resistant. So we can't induce brain inflammation to then follow the fate of monocytes across the brain, because nobody moves into the brain, or whatever you have is so skewed and so weird, there's no inflammation. And equally and very paradoxically, and I hope nobody asked the question to what does interferon gamma do? The answer is, I really don't know. Strangely enough, we all know and think of interferon gamma as one of these quintessential pro-inflammatory cytokines. But a mouse that does not make interferon gamma, or a mouse that does not have the interferon gamma receptor, when you induce EAE, will get much sicker. So it is not that interferon gamma is driving inflammation, but somehow it seems to restrain inflammation in the brain. Again, I don't know why, but moreover, these mice are not useful for our purpose here. We can't use a mouse where there's no inflammation, and you can't have a fair comparison between mice, between cells. One is prone to leave normal high inflammation, and the other one goes to crazy next level inflammation. We needed something more equal. And we did something that we haven't done in a long, long time, something very simple and silly. We made mixed bone marrow chimeras. We're very careful to create mice in which 50% of the monocytes are wild type and can sense both GM-CSF and interferon gamma, and 50% of the monocytes are either blind to GM-CSF or are blind to interferon gamma. We let these mice rest for a while, and then we induce EAE. So this is basically the scheme here. Super, super simple and primitive. And you can see here we're very careful that in the peripheral blood, we really have this 50-50 chimerism. And then whatever we see in the brain, we can recognize a cell that has sensed GM-CSF and has sensed interferon gamma, or has sensed neither of them or both of them at the same time. So we could do this experiment with this scheme here. And here comes one of the things that I thought was quite interesting for us, is that interferon gamma gave us a rather dramatic, this is the composite here of a mixture of this is what happens to monocytes, the labeled monocytes that we had when they can sense both interferon gamma and GM-CSF. This is what happens to monocytes when they can't sense GM-CSF, and this is what happens when they can't sense interferon gamma. Now, there are these many clusters that we put here. I would love to say that I do not want anyone to believe that there are 10 different clusters of monocyte-derived cells in the brain. This is a purely mathematical approach. I mean, I see this sometimes when people say, oh, and then we clustered this. This is not really the purpose of this experiment, that we identify new subcell types. This was not the purpose at all. For us, it was more to see the gestalt and the behavior of monocytes when they can't sense GM-CSF and when they can't sense interferon gamma. But these clusters are not necessarily very, very meaningful biologically beyond our ability to capture what is missing. And so what you may be able to see here, let's see, is that, let me just label this up here, is that in GM-CSF receptor deficient monocytes, we see a particular loss of these three clusters. And these three clusters that we see here, so that's missing here, are primarily responsible for the production of reactive oxygen species, of the inflammasome, and in phagocytosis. These were important. And again, this is just phenotypic so far. Do we know that this translates into reality? I'm not a huge fan of leaving it at single-cell RNA-seq data. I still think you need to have some sort of functional verification that this is happening. And unfortunately, this took us much longer than I thought it would take. But basically, what was able to basically, when we looked at these three features that I said, inflammasome, reactive oxygen species, and phagocytosis, yes, indeed, in the absence of GM-CSF sensing, monocytes will make less IL-1 beta, will make less reactive oxygen species, and will be less efficient phagocytosing. Often be used here. We used, I think, just beads were used for this as a test. So let me just summarize this, and this is basically the end of it already. I promised I would rather browse through it a little bit quicker, so we have a bit of time for discussion. In tissue inflammation, in this case, in neuroinflammation, the system is geared towards the production of interferon gamma and GM-CSF, two cytokines with both a major effect on monocytes and monocyte-derived cells. And so what we found relatively clear is that Ly6C high monocytes will upregulate MHC class 2. It's not the first things that we see, and they will then develop into monocyte-derived cells, and then in the brain. And later on, they will become monocyte-derived macrophages, predominantly expressing reactive oxygen species, interleukin-1 beta, and they will phagocytose. This transition here is really dependent on interferon gamma, and it's fundamental how massive the effect of interferon gamma is phenotypically, and yet we don't quite understand the function. But GM-CSF is a more subtle effect that has a much bigger impact on function. So phenotype with interferon gamma is bigger, and with GM-CSF the function was bigger. As a side note, without wanting to confuse issues here, there is an interesting phenomenon happening with this transition here, and that is in the lymph node. I didn't go into this so much, but in the draining lymph nodes, monocytes invade, will make interferon gamma, will sense interferon gamma, will upregulate MHC class 2 molecules, and moreover, actually, we have data that are coming out in this paper showing that this event is actually important for T cell priming in the lymph node. It isn't important in the brain anymore, because when you delete the MHC class 2 molecules in the fully inflamed brain, it has no effect whatsoever. Incidentally, there was a paper recently by Alex Miltner and colleagues, a very, very interesting paper showing the presence of these CXCL10 positive monocytes, like a super subset of monocytes that we weren't able to study as to whether they are super important or not for brain inflammation, but they were not affected very much by interferon gamma and GM-CSF. And I think with this, yeah, this is basically the last picture I've taken with the team, and I think this may have been 2017. Sadly, we have not been able to come together as a lab since a very long time in order to, let's say, take such a picture in an intimate way. So we would have to wear masks right now. Obviously, same is true for you. And because of rising numbers, we have our famous annual Christmas party, which basically goes pretty much completely mad with too much food, too much alcohol, and usually lasts until the early morning hours, may have to be canceled yet again. And I'm starting to be rather depressed about that. But I would like to, the individuals who have contributed to the work, I've mentioned them. And on the left, you see our collaborators that I need to thank for this. And I'm sorry if at one point or another in this presentation, I made a few mistakes and I have to move slides around. If we see each other, if you ever see this presentation again, I'll do better. I'll correct it just after our talk. And with this, I would like to give back to Juan.
Fantastic. Okay, well, I mean, that's really great. I guess, so I think before the Q&A section, I want to double check with you how long you have for us to ask questions.
Well, I don't know. So I roughly calculate. I mean, I don't want to be. So I'm going to level with you. I'm buying an old farmhouse in Milano, in the area of northern Italy, not in Milano, but I was hoping I could go there this afternoon. So for me, it's right now 2:45. So I thought we'll talk as long as we want to talk and chat, and then. But I'm happy to also engage with anyone individually at some other time as well. So if we really have more than 15 minutes of Q&A, we can extend without any trouble either today or some other time.
Sounds great. Okay, well, let's start it. I think the first question from Hilal. Do you want to ask a question yourself? Go ahead.
Yeah, sure. Are you getting me?
Yeah.
Yeah. So I was wondering about the states of MDSCs there. Do you think that in case of induced model of GM-CSF, there is the chance of expansion of MDSCs there? Because we know that GM-CSF is a very much potent growth factor for the promotion of MDSCs there. So do you think that it can promote a reverse effect, that is, it can promote the anti-inflammatory effect there?
Yeah, so it's an excellent question, and one of the questions we are asking in the lab ourselves as well. So we built quite a few tools around GM-CSF in order to address this particular question. The question, in other words, is how important is GM-CSF for the formation of, let's call them tumor suppressive macrophages? And the answer is I don't really know the answer. There is maybe no general answer like yes and no. GM-CSF drives regulatory functions in macrophages or not. It is much more context and tissue dependent that we already know. And it also is very, very important as to what is the origin of the TAM. Is the TAM a tissue resident cell, or is the TAM coming from the systemic immune compartment like a monocyte? Then you also have other cells such as neutrophils that are also GM-CSF responsive. So this whole gemisch makes it really, really difficult to give a unifying answer. All I can tell you is I think that it is complex and we're getting there, but we have to tease one cell apart from the other very systematically, and it takes a bit of time. But it is one of the most pressing questions with great relevance actually to cancer. It's a very good question. Thank you.
Okay, so let's continue. I'm now reading out the question in the chat box. So one of the question is, in the event of loss of GM-CSF producing cells in the central nervous system, would this result in M-CSF overcompensating?
Ah, that's a good question. So it's difficult to model that because, I mean, if you do not, let's say you have a GM-CSF knockout mouse, the problem is you don't get inflammation, and hence you cannot see what's the contribution of CSF1. So that cannot be done in this way. So the question would more be, what happens in the central nervous system once you have neuroinflammation and you shut down GM-CSF? And we sort of did the experiment. We did do this, the suicide frog experiment is such an experiment where the moment GM-CSF is produced, the cells will commit suicide. And so what we observed there is dramatic loss of inflammation. And we looked, if I recall, specifically at interleukin 34, another CSF1 receptor ligand. And if I recall correct, did not see a major increase in its expression. And I don't recall the data for CSF1. So I don't know. It's a very good question because you're right. Obviously, there is something about it. Nevertheless, I should add that I personally do not believe that CSF1 can compensate very well for the loss of GM-CSF in terms of function. It can help to sustain cells. It can potentially help to make cells stay alive and to be maintained. But I do not personally believe that the data is strong, that it can drive this pathogenic program. This is really rather specific to this GM-CSF STAT5 phosphorylation mechanism, which is not so overt in CSF1.
Okay, so Hongyan, you want to go ahead and ask a question?
Yes.
Go ahead.
Thank you very much. This wonderful talk. I have two questions. First one, I think GM-CSF is very important, but it cannot work alone. So what kind of other key cytokines or other neurotransmitters could work together with GM-CSF during your neuron inflammation?
Okay, so this is something we can answer to some extent. So I can tell you that this monocyte to phagocyte transition is really dependent on interferon gamma. So interferon gamma is really needed for that. Nevertheless, and here comes the kicker and the complicated thing, even if you abort this phenotypic transition of a monocyte into a monocyte-derived, what have you cell, monocyte-derived cell that has adapted to the local microenvironment. Even if you interrupt that, if GM-CSF present, you can still trigger this pathogenic program. So I am, like you, convinced that other molecules are playing in concert together with GM-CSF to act on myeloid cells this way. However, I do not think that any of them that we've looked at has a non-redundant function, whereas GM-CSF is non-redundant, so we really need that. The question that makes the whole thing much more complicated is that the production of GM-CSF in T cells, we thought we had that sorted out exactly how that's regulated, but the cytokines that influence this and drive the production of GM-CSF in vivo, we don't really understand too well. We have been trying for quite a while to get a grip and to understand, but so far we really struggle to I can't answer the question very well. We know IL-2 is important, but that's it in a nutshell.
Right. Okay. Thank you. My second question is about microglia is the local macrophage in the brain. So could the GM-CSF affect microglia similarly as what it did to the monocyte?
That's an excellent question. And I have to say, so first of all, yes, GM-CSF can be sensed by microglia 100%. Microglia, the moment GM-CSF is present, microglia have the receptor and they can sense it. However, it has no impact whatsoever on the disease. So we have knocked out the GM-CSF receptor specifically from the microglia compartment, and you still get perfectly fine EAE. There's absolutely no trouble whatsoever. So the microglia, which was the first suspect that we had, are not GM-CSF dependent disease drivers at all, for sure.
But I have one argument. Even if you delete microglia, you still have monocyte or macrophage driven from the blood. That can
so we didn't delete microglia. No, we have the microglia present. So in our case, you're right. If you delete microglia, you, depending on how you delete microglia, you either have a non-killed microglia proliferating and moving into the cellular niche where the other microglia were, or depending on how you set up the experiment, there's another way, there are a couple of papers on this already, you get indeed monocytes moving into the central nervous system and looking very microglia-like after some time. What we did specifically was we did not touch the microglia. The brain is full of microglia, but the microglia are blind to GM-CSF, so they can't sense GM-CSF. And the only cells that can sense GM-CSF in the system indeed are then the monocytes, and those are the ones that must sense GM-CSF. So it's the monocyte-derived macrophage is important. The microglia, by origin of a yolk sac macrophage, those microglia, the brain resident cells, do not contribute to this to the GM-CSF-driven pathogenesis.
Okay, thank you.
Sure.
Okay, so Xingyuan, you want to go ahead?
Yes, can you hear me?
Yes, I can.
Okay, great. Dr. Becher, this is Xingyuan Fu. Probably, we have talked before.
Yes, I remember very well. We met. Yes, of course. How are you doing?
Good, good, good. I really love your talk. And
oh, thank you so much.
The wonderful work.
Me too.
Actually, I
thank
circulated your talk in my lab here, and also that's congratulations. That's so dedicated work.
Thank
I would
you
say
so much.
This is extremely, I think, is a high quality work you have done too. So
thanks.
my
means
question
a lot
is very
to
simple. Yeah, I also thank you for quoting our paper in your papers, whatever you published on the first observation. This so we call it THGM. I think you agree with this name, right? It's a T helper cell producing GM-CSF.
Yes, to some extent I do. While there's no question about the nomenclature is possibly spot on to call something THGM, but I'm starting to turn around and say, like, maybe I actually don't want people to call things TH1, TH2, TH17, TH9, TH22, or THGM anymore, because I really would like to, I personally think it's more useful to condense it. Because as you know, THGM cells are often also intermingled with traditional TH1 cells and so on. And sometimes IL-23 is also important for the GM-CSF production. So we see cells that make IL-17 and GM-CSF together. So I got so confused. I'm trying to boil it down to so that we don't have like 15 different subpopulations of THGM cells.
That's in some way I agree too. But in this case, particularly showing the EAE case, I so amazed to see this also GM-CSF is a serial driver, not other cytokines.
You're right.
That's really, really impressive. Yeah, too. Yeah. But I would like to ask you, because we showed also other group had showed that all these TH helper cells, it's driven by one of the STAT members, like TH17 by STAT3 and TH1 by this STAT4 and so on. So how about this? Have you ever looked at, we at least to show STAT5 is important for this GM-CSF expression. And
absolutely.
Yeah.
No, the short answer is we did look very carefully. In fact, we use STAT5 phosphorylation to test our anti-GM-CSF antibodies. We use STAT5 phosphorylation to see whether on the single cell level as to whether we knocked out the GM-CSF receptor, for instance. So GM-CSF receptor, as you rightly say, absolutely signals through STAT5, and it's a non-redundant signal. You're absolutely correct. And I like the idea also to think about maybe what would it look like if we think about translating the function of helper T cells based on the different stats. That's actually a very interesting concept that we haven't developed much, but it's a very, very interesting idea as well.
Thank you for telling me that, because recently I have a bet with Chen Dong. If you know, he's one of the discoverer for TH17. He also in China. So I just make a statement with him saying, whether in the EAE model, because he insists that still TH17 is the driver. But I said, look at Dr. Becher's work. I even sent him the paper, your paper. I said, it's not our work. It's also your very, very delicate, fine genetic evidence. I think that's kind of be better even too. I thought this system you designed. So I sent it to him. He never replied me. Anyway, so that's anyway, the scientific dispute is normal. But I would like to tell you, we all now is making inhibitors. Actually, I'm setting up a drug company, basically using the CHGM as the major targets. We also patent all this in the almost 7 years ago now, since our paper published in 2014. Now this already endorsed also in the Europe, United States, and also in the
Congratulations.
China. So that's, I hope really someday you can come to China, visit us. I even have a number of paper on my desk, but I don't have time to publish it. And now we are busy with the drug making, and hopefully this discovery, including yours, this GM-CSF, I'm glad to hear the STAT5 is indeed being a driver for this GM-CSF expression. So that's our major target. We are trying to making a STAT drug, actually. Now the GM-CSF antibody is already available, particularly by my friends, and you probably know him. It's called Dr. Zhang Jingwu Zhang, and he used to publish something on the TH17. And but now his company also very aggressive making the GM-CSF antibody. So I hope his antibody going to contribute to these patients. I just make something for him because I don't know he knows your presentation or not. But that's I would think that's great. Probably if you need a humanized GM-CSF antibody, I can help you.
Okay.
Talk to Jing Wu Zhang. Yes, excellent antibody. Yeah.
Thank you very much.
Anyway, that's quite great. Let me give the time to others. Thanks a lot. I hope you come visit the channel. We can video chat sometime. I hope. Do you have a WeChat?
I do actually.
Oh, that's wonderful too. Maybe I will email you. You send me a WeChat account. That's we
Sure
can
thing.
talk WeChat. Yeah.
Absolutely.
I also thank Dr. Wu Chuan to host this great seminar. Actually, my lab all joined it, enjoyed. Thank you again. Okay.
Okay, so I think there's a lot of, I don't want to hold you too much, Becher. I just, I think last question I just put out there is people ask if the GM-CSF thinks it's pathogenic cytokine, which which probably not only focus in the central nervous system inflammation. What about this cytokine in other related autoimmune diseases?
Yeah, that's a great question. I like it a lot, and the answer is it's an emerging field, right? So in the context of rheumatoid arthritis, which is where it started, basically, GM became much more popular in RA than in other disorders. So that's one of the first indications, and a phase 3 trial has been concluded, hasn't been published yet in detail, but it may very well be that GM neutralization will become very, very popular for rheumatoid arthritis. So that looks very, very good. There is a clinical trial that I'm very excited by because we're a little bit involved in that. It has to do with the treatment of graft versus host disease. And I'm very excited about that, so that the neutralization of GM-CSF may alleviate GVHD. And if you've ever seen a patient with GVHD, this is a very worthy cause. It has not worked in psoriasis, and for reasons I don't understand was even tried there, because I think there was no evidence to try GM-CSF in psoriasis. It also doesn't work in the animal model for psoriasis, so it's just one of the things. It isn't a universal driver for every inflammatory type, but what I call these quintessential type 1 inflammatory lesions are there. I think GM-CSF has a place.
Okay. So I told you that you will get bombed with the questions. So, I mean, people are so interested in what.
Let's go on. That's fine. Let's have a few more questions if you like.
Okay. Let's say do a few more then. One question is Th17 is critical in EAE development. Is GM-CSF mainly produced by Th17 cells? And have you ever checked IL-17 or Th17 in your GM-CSF deficient model?
Yes. So in the absence of GM-CSF, again, inflammation is gone, so it's not easy to study other cytokines then. When we lose GM-CSF producing cells, we have IL-17 producing cells. Now, I personally know that IL-17 has an effect in EAE. There are two different effects that I think are very prominent. One mentioned earlier, that IL-17 has an effect on the endothelium, and in particular, and there's some very strong data out there that the blood-brain barrier function of the neurovascular unit is inhibited, or is changed through IL-17. And there's a recent paper by Ari Weissman in Science Immunology saying that that IL-17 is very important for the function of the composition of the gut microbiome, and that the gut microbiome instead is very, very important for the fitness of encephalotogenic T cells. So this is basically what I think is happening. So, but I don't see how IL-17 would directly kill oligodendrocytes or something like this. I don't think there's good evidence for this. But there's no question about it that mice lacking IL-17 have a phenotype in EAE. But the reason as to why that is still a matter of debate. And your question as to whether GM-CSF can be made by Th17 cells, and the answer is it's hard for me to say that, but we do see cells that make IL-17 and GM-CSF. Yes, we do. There is also this interrelationship with interleukin 23, which clearly has, it's not so important for IL-17 production but it's very important for the pathogenicity of these IL-17 producing cells in the context of neuroinflammation. And we think that that may very well be because of GM-CSF, but that's all I can tell you at this point.
But do you think that GM-CSF influence the IL-17 producing cells?
I don't think so, because the answer would be no. I'm sorry. To be very precise, directly, no, that's not possible, because T cells do not sense GM-CSF. They cannot see it themselves. They can only make it, but they can't be influenced by it directly. But indirectly, obviously, yeah.
But I think I want to ask you insight about Tbet actually being known control GM-CSF, which actually important for both Th17, pathogenic Th17 and Th1 cells. So do you think that the IL-17 producing or IL-17 generation and GM-CSF generation are sharing the common signaling pathway?
Again, I don't know the answer to that. I wish I could find out easily, but we don't see a very, very important Tbet signature in GM-CSF producing cells. But you're absolutely right. Tbet deficient mice will also have less Tbet, but the GM-CSF. The question is more, why is that? Is that a direct effect of Tbet on GM-CSF expression, or is it another transcription factor that may be driven by Tbet that can then be engaged for GM-CSF? Those networks are really, really complicated. We're trying to find out. Actually, that's one of the reasons I'm going to Italy today, to meet with some of my colleagues there who have much more experience looking at transcription factors and how they're regulated. And it's one of the goals to collaborate with them and find the answer to your excellent question.
I think next question from Sandong. You want to go ahead? Okay, well, I think Hongye want to ask one more questions. Hongye, you want to ask?
Yes, actually,
Okay.
I'm very, yes, can you hear me?
Yeah.
Okay, I really enjoy your work, so I want to ask the final question. How could GM-CSF affect RS production, IL-1 production, and also phagocytosis together? I know that the single cell sequencing data are very interesting. You see these all three subsets are missing in the GM-CSF receptor knockout mice. Yeah.
Yes. That is basically the best answer I can give you. I do not know how GM-CSF can regulate such diverse processes and what the transcriptional network is yet. And it'll be difficult to find out, but it would be very interesting to find out. Nevertheless, the data is I think is rather compelling, because you do have GM-CSF sensing and blind monocytes next to one another, and the ones that can sense GM-CSF will activate the inflammasome, will make reactive oxygen species, and will be stimulated to make phagocytosis. And the ones that don't sense it next to it will not behave this way. So I don't know the details of how to trifurcate, let's say, these functions, and whether there's overlapping signaling or not. I would assume there is, but I do not know the answer. How that works remains unclear.
Right, but when I read some references, if autophagy or phagocytosis is activated, usually inflammasome activation is inhibited. So they are not both activated. So that's why I have this question.
So did you say autophagy or phagocytosis? Yeah, so we did not look at autophagy at all. So for us this year is really phagocytosis and not autophagy that we're looking at. We're really looking at the active process of proper phagocytosis, not macropinocytosis and not autophagy. We only looked at the ability to eat, and that is enhanced in the presence of GM-CSF, and that's one of the things that was reasonably well documented before our study, actually, by others as well. So but the effect of GM-CSF on autophagy, I don't know. I have no idea. I believe it. I assume you're right. If the inflammasome inhibits autophagy, then I would assume autophagy is decreased, but not phagocytosis.
Okay. Thank you very much. Thank you.
Sure.
So you see, after 30 minutes of talk, it's still a lot, 100 people online waiting your, you see my question, I have a list of questions. I don't want to hold you too much. I have one last question. So
Sure.
I'm actually curious about this GM-CSF tissue specificity. And can you actually provide some insight on that? Because I noticed that you're describing that GM-CSF is so important in the central nervous system, but at the same time, you also mentioned there's no so much impact in the peripheral. I'm curious,
Correct.
what do you think?
Okay, so that is basically the gist of the question. This is a very, very good question. It has to do with this model of dysregulating GM-CSF. So we dysregulate GM-CSF in the periphery, and then at one point, boom, we see this crazy brain inflammation and influx of monocytes into the brain, and then the brain is eaten up. So how is that possible? And I do not think that this is a very clever directed procedure of monocytes saying, "Ooh, there's GM-CSF, I have to get into the brain." I don't think that's what happens. I think what happens is you open the valve of the monocyte bottle, and now they're getting out of the bone marrow at rather large numbers. So you do have this strong myelopoiesis going on. This leads to this excess of monocytes everywhere. So in the systemic immune compartment, they're everywhere, right? Many, many more than you would usually have. And they're able to visit things like the lung and the kidney and the liver, and they visit these tissues on a regular basis. And that doesn't seem to be such a big deal. So the lung, the liver, and the kidney seem to be able to handle the visit by monocytes. But at one point, when too many monocytes visit the brain, and I think that's basically what it is, when the floodgates are open, too many of these cells are coming in. I think the brain is rather xenophobic and is not particularly happy by these newcomers from a very, very foreign place that start to eat and everything. So the brain is not able to cope with it. That seems to be the problem more. I don't think this is a directed attack against the brain at all. I think it's a coping mechanism of a very weak organ when it comes to dealing with foreigners, if you wish.
We're back to the US analogy.
I didn't say that.
Well, that's excellent. I mean, thank you so much. Let's see. Anyone else want to ask questions? I don't want to hold Burkhard too much from travel.
No, but you're also welcome to. I mean, I'm happy to. If somebody says, "Hey, let's have another meeting together," or so. I couldn't come, I couldn't visit, obviously.
Yeah,
but if somebody says, "Hey, I would like to have a quick chat with you," or send me a quick email. I did this lately. There was another thing that might be interesting for you and this activity. There's the global immunotalks, which I'm now helping with. I'm very excited by this initiative. And what we do is at the end of the presentation, people then go to Twitter and ask the questions by Twitter. And the presenter has to answer by Twitter too, which I think is actually a very cool initiative. And it might be, so if you want to do that, you're welcome to either email or tweet me, and I'd be happy to share thoughts and data. Absolutely. Yeah, we should become Twitter followers. I don't know whether we did that already. All right, let's do that then.
Thank you so much for doing this.
Thanks for having me.
Yeah, absolutely. It's so great to see you again. I mean, hopefully, we're gonna be able to see each other face to face. So
I would like that. Thanks. Likewise. It was good seeing you.
Okay,
take care, everyone.
Bye bye.
Thanks for your interest. Bye bye now. Bye.
Bye.